Hello, welcome back guys. It's Ryan Lim again and we're in part 2. Okay, the fourth chapter of the part 2 is molecular genetics. So in here we're going to talk about the DNA replication and DNA expression. Okay, so DNA replication is talking about the copying of the exact same DNA from the original DNA, and the DNA expression is talking about how you make proteins out of the DNA information. So the first chapter, the molecular genetics, consists of the 3 sessions. In the first session, we're going to talk about DNA replication only. And in the second session, we're going to talk about the DNA expression. And in the third session, we're also going to talk about something that we didn't talk in the basic level, which is the viral genetics, the DNA of the virus, and also bacterial genetics, and some, the experimental techniques. Okay, let's get to the first slide. Before we get to the real material, we're going to briefly talk about the basic terms. So genetics is a study to understand the processes of the inheritance, right? So basically studies how DNA is replicated and expressed. And the genome is a term that refers to the entire sets of the genes, right? So all of your genes make one genome, and every genome is different for every organism. All right, and to make genetic information, you need to have 4 different bases, and which is A, T, C, G, right? So they make different number of the hydrogen bonds like this. So that's how they can have their specificity, right? So A specifically pairs up with T, and G always pairs up with the C, right? So A and T, you can find 2 hydrogen bonds. G and C, you can find 3 hydrogen bonds. So if A is trying to make up a hydrogen bond with the C, they're going to make like 1 or only 2 hydrogen bonds, right? Which is much unstable than when it pairs up with diamine. Or if G is trying to pair up with the diamine, it might be able to make up only 1 or 2 hydrogen bonds, right? So in that case, that's also unstable. So A always pairs up with T, and G always pairs up with the C. All right, like this, A and T, 2 hydrogen bonds, C and G, 1, 2, 3, 3 hydrogen bonds. Okay, so for the genetic processes, we can have 2 different things, right? For the DNA, we can duplicate them or replicate them, or we can express them. When you say gene is replicated or duplicated, you just make exact same copy, and that only occurs before the cell division, either mitosis or meiosis. Okay? And for the genetic expression, you synthesize polypeptides or protein using this DNA information. Okay? So the expression pattern determines different types of the cells in multicellular organisms. So think of the human body. So we all have, entire cells of the human body contains the exact same DNA for that specific individual, right? But still, we have all sorts of the different types of the cells, such as the heart cell, muscle cell, or our gonad cells, or the brain's neuron cells. So that means even though we have exactly the same DNA sequence, by controlling or regulating our DNA expression pattern, we can make different types of the cells. Okay, so before we get to the DNA replication process, we got to talk about the directionality of the nucleotide first. So if you look at the one nucleotide here, you can have a base, sugar, phosphate, and obviously you can find which one is top and which one is bottom. If you say this one is top, this one's got to be bottom, right? So you can definitely distinguish this part from this part. That means your nucleotide has directionality. Same for the nucleic acid that contains more than 1 nucleotides connected by phosphodiester bond, right? So here, if this one is top, this part has to be your bottom. And when you have a double helix, so in this case, you just have one strand, single strand. But if you have two strands, they can make complementary base pairs all together, and they become one double-stranded DNA. Okay, so SS stands for single-stranded, DS stands for double-stranded. And one of the most important features is these two strands are in the opposite direction. Okay, so that's why your double helix is called to be anti-parallel. Okay, anti-parallel. That is because if you have one double-stranded DNA like this, okay, we just unwound this double helix, so that's why it looks like a letter shapes. And if the directionality from here to here is, if this one is the top, if this one is bottom, and the opposite strand, this part has to be bottom, and this part has to be your top. Okay? In other words, for the top part, we usually say that is our 5 prime, and for the bottom part, we usually say this is a 3 prime. Okay, so we have one sugar that contains 1, 2, 3, 4, 5 carbons, right? So, and you can only label the numbers on your carbons, not on the hydrogen, not on the oxygen, right? So total 1 prime, 2nd prime, 3rd prime, 4th prime, and 5th prime. So this top part is 5th prime or 5 prime. Bottom part is your 3 prime. So if you draw this anti-parallel double helix again, you usually indicate the directionality with the arrow. And if this part is 5 prime, this part has to be 3 prime. And since it is anti-parallel, this part is 5 prime, and here it is 3 prime. Okay. It is totally very, very important to understand the DNA replication process. Okay, so we have a hydrogen bond in between the base pairs, complementary base pairs, AT, CG, GC, and the TA, and you have anti-parallels, opposite direction, 5 prime to 3 prime, 5 prime to 3 prime, right? And you can see here the orientation of the ribose, orientation of the sugar group is different for this one and for this one. Okay, so if you wrap up and summarize all the characteristics of the nucleic acid, 2 most important things, double helix and anti-parallel. For the DNA replication, semi-conservative. I believe you all know what it means. So when there's one double-stranded DNA, after DNA replication, so you basically split it up in two single-stranded DNA, so this single-stranded and single-strand DNA, and then you do the DNA synthesis, polymerization, right? So each single-stranded DNA can get complementary base pairs laid down according to their sequence, so you can make 2 complete double-stranded DNA, which is exactly the same. Okay, so since you use only half of the strand, or since you can find only half of the original strand from one complete newly synthesized double helix, you call this phenomena semi-conservative.